and it's my great pleasure to introduce our first speaker this morning, who is Mike Stratton. Mike, of course, is the director of the Wellcome Trust Sanger Institute, and of course, Mike's primary research interest has been in cancer genetics, and he's well known for a number of research discoveries, including early work on the BRCA2 gene, and then, of course, subsequent series of studies in several areas on cancer risk and cancer susceptibility. Mike, of course, also initiated the Cancer Genome Project at the Wellcome Trust and focuses his studies in an ongoing way on understanding somatic mutations in cancer genomes and their implications. Mike is very well recognised, of course, for his groundbreaking work with a number of accolades and prizes, including, of course, his election as a Fellow of the Royal Society, and in 2013, his receipt of a knighthood. It's a great pleasure to have you with us, Mike. Welcome to WEHI, and we look forward to hearing from you on signatures of mutational processes in human cancer.
Thank you very much for that generous introduction, and thank you for inviting me. It's really a great honour to be here at the WEHI Centenary. It's a great honour to be invited, but it's also a great honour to convey to you the respect with which this institution is held around the world, respect for the research that it has done in the past and continues to do today. So most of us subscribe to the notion that all cancers are caused by somatic mutations. However, what is slightly surprising and remarkable is actually how rudimentary our understanding is of the mutational processes that cause those somatic mutations, because those mutational processes are actually the causes of cancer. So at a very high level, we know that all cells in the body acquire somatic mutations throughout the course of life. They acquire mutations every time a cell divides and has to replicate the 6,000 million letters of code. We know that every cell in the body is exposed to mutagenic influences throughout the course of life. The intracellular environment is actually quite a toxic one, and there are tens of thousands of damaged bases in every cell, and those damaged bases have to be repaired every day, otherwise they get converted into somatic mutations. We know that if we expose ourselves in various ways, for example, to radiation, that will cause DNA damage in the tissues that are exposed. That DNA damage will be converted into somatic mutations, and that may cause cancer. Similarly, if we ingest various carcinogenic mutagens, they will cause DNA damage in tissues which are exposed, causing mutations, ultimately causing cancer. Some of those carcinogenic exposures we cannot avoid. This, for example, is aflatoxin. This is produced by a fungus, an aspergillus fungus that infects maize in certain parts of the world. And if one consumes the maize, one consumes this stuff, which goes to the liver, causes DNA damage, causing mutations, causing liver cancer. So the DNA in our bodies and every cell of our bodies is continuously under assault. And therefore, there are a suite of mechanisms that are operating every day in every cell of our body that are there to repair that DNA damage. And if a component of those mechanisms begins to malfunction, then that particular cell will begin to acquire more mutations than it would like to, and that will configure that cell ready to become a cancer. So it trips off the tongue easily to say that ultraviolet light, excessive exposure to ultraviolet light predisposes to the development of skin cancer, or that ingestion of tobacco carcinogens predisposes to the development of lung cancer. However, if we ask the question, what are the causes of the mutations that cause breast cancer, or what are the causes of the mutations that cause glioma, or what are the causes of the mutations that cause pancreatic cancer, we have very little understanding of that. So perhaps we can get some idea of those mutational processes from studying the cancer genome itself. And the first indication that this could be the case came from a suite of studies by many investigators in the 1980s and 90s looking at p53 mutations in human cancer. And the mutations that we're going to be considering, in the first instance anyway, are just base substitutions. And there are 6 different types of base substitutions: C to T, C to A, C to G, T to A, T to C, and T to G, if one starts with the pyrimidine of the Watson-Crick base pair. So if we look at p53 mutations, for example, in skin cancer, what we find is that most of those p53 mutations are C to T substitutions. And that makes a certain amount of sense, because we already knew from experimental studies that ultraviolet light, which causes skin cancer, also causes C to T mutations in experimental systems. Similarly, if we look at the p53 mutations in lung cancers, we find now that there is a large proportion of C to A mutations, and this also makes sense because tobacco carcinogens cause C to A mutations in experimental systems. So in other words, what the studies of p53 were telling us was that the cancer genome, in this particular case the p53 gene, bears the mark, it bears the imprint, it bears what we now call the signature of the exposures that were involved in causing those mutations in the first place, those mutations which ultimately led to cancer. So can we now use the thousands of cancer genomes that have been sequenced in order to get a much broader view of the mutational processes and exposures that are implicated in developing all types of cancer? To do that, we need to think about a little bit about what we think is happening. This is a single cell from a late-stage cancer. These are the somatic mutations in the genome of that cell. And for that cell, as for any other cell in the cancer or any other cell in the body, we can construct a lineage going back all the way to the fertilized egg. So this is a lineage of cell divisions, and I'm showing you some snapshots of cells within that lineage. Where the cells are colored brown, they have already undergone neoplastic transformation. They're also already cancerous or partially so. And where the cells are blue, the lineage is going back through normal tissues. And what you can see is that the somatic mutations are being accrued over the course of a lifetime in an incremental fashion, and somatic mutations, this one here, for example, which may have been generated at the first division of the fertilized egg, is still there in this late-stage cancer cell 70 years later. So when we consider now the mutational processes that have generated those somatic mutations, let's consider first this hypothetical mutational process A. Here are the 6 different types of mutation, and this particular mutational process is leaving a signature characterized predominantly by black mutations. It doesn't matter what they are. Now, this particular mutational signature and process may be operative all the way through life, but what you can see is that from the thinness of that line, that the exposure is rather weak throughout the course of life. So that's just one hypothetical mutational process that is operative throughout the whole of that lineage, generating the mutations in that final cancer cell. There may be another one that's operative. This mutational process B leaves a mutational signature characterized by red mutations and purple mutations predominantly. This one may be operating only through a portion of that lineage, but you can see that when it does operate, the intensity of exposure is great. And for example, tobacco smoking could be an example of that sort of mutational process. And there could be a third hypothetical mutational process that is operating throughout the lifetime of this individual and throughout this cellular lineage. This one is characterized by a mutational signature with mainly green and orange mutations, and this one may start relatively late on, after neoplastic transformation has started, and go right through to the end. And there could be more processes operating. But the point is that throughout the course of a lifetime, these 3 and perhaps other processes are all operating to make the mutations that are present in that final cancer cell. And when you look at the mutational catalog in that final cancer genome, you can see that it bears relatively little superficial similarity to any of these three mutational signatures or mutational processes because they've been all added up together, they're all jumbled up together. And our challenge now is with thousands of catalogs of somatic mutation from cancer genomes looking like this, what we want to do is to go backwards and extract out these mutational signatures that contributed to those mutational catalogs. And to do that, we have to make a reasonable assumption, which is that actually, if we look at different individual cancers, the contribution of these different mutational signatures and other mutational signatures will differ between individual cancers. And if that is the case, then we can reduce it all to a set of mathematical equations. There are a number of different ways of solving those mathematical equations to extract out these mutational signatures, but one of them that particularly lends itself to this process is called non-negative matrix factorization, and it's been used in many different contexts. This is one shown here. Here, it's been used to extract out information from digitized images of faces. So a lot of faces shown here, and what the process does is it extracts out the components of faces, eyes, mouths, noses, and so on, exactly in the same sort of way that we want to take thousands of cancer genomes and extract out the components of the mutational processes and signatures that are there that are contributing to those cancer genomes. So that's what we're going to do. But before we do that, we have to revisit our classification of mutations. Because this classification, according to six base substitutions, it doesn't give us the ability to resolve the mutational processes in the way that we want. It doesn't have high enough resolution because we already know that there are several mutational processes, for example, that cause C to T mutations, and we wouldn't be able to distinguish between them unless we had a higher resolution classification of mutations. So to do that, we look at the bases either side of the mutated base. So here is a C to T substitution, and you can see that the base before is a T and the base after is a G. So this is one subtype of C to T mutation. Here is another subtype where the base before is now an A, and here's a third subclass of C to T mutation where the base afterwards is a C. So there are 4 bases immediately prior to the mutated base and 4 bases afterwards. And therefore, for C to T, there are 16 different subclasses of C to T mutation. And if we look at all 6 different base substitution, there are 96 mutation classes. And this is the classification of mutations that we're going to use. And this does give us the resolution we need to distinguish between a large number of mutational signatures, as you'll see in a moment. So this is the recent version of the analyses that we've been doing. We've taken over 12,000 cases of cancer, which have been sequenced, and somatic mutations have been extracted from 40 different cancer types. So that's 12,000 mutational catalogs of, in a total, 8 million somatic mutations. We then apply that framework, that mathematical framework of non-negative matrix factorization. And what that does is it extracts out the mutational signatures that we want to look for. And in addition, what it'll do for us, it'll estimate the contribution of each mutational signature to the mutational catalog of each of those 12,000 cancer genomes. So using that approach and that classification, we now can look at the landscape of mutational signatures across human cancer. And this is how I'm going to show you a mutational signature. You can see the 6 different base substitutions here, and underneath each of them, there are 16 possible bars indicating the sequence context in which the substitution is occurring, giving you a total of 96 possible bars across the whole figure. So this signature is characterized predominantly by red mutations, which are C to T, and you can see that they are occurring in 4 particular sequence contexts. So that's how I'm going to show you the signatures. So the landscape of signatures that we have found across the whole of cancer thus far includes 30 signatures shown here. In a more recent analysis with more whole cancer genomes, it's up to about 35 different mutational signatures. In other words, 35 signatures, 35 underlying mutational processes. So the first message really is that there are a lot of mutational signatures and therefore processes underlying the somatic mutations in human cancer, perhaps more than most of us were expecting. They're also pretty diverse. Here you see the two extremes of diversity. In this one, signature 3, the mutations are distributed in a rumble across the 96 different potential mutational types. At the other extreme is this signature here, where 2 out of the 96 subtypes of mutations account for most of the mutations in that mutational signature. Many of the signatures are indeed composed of red C to T mutations predominantly, some of predominantly blue C to A mutations, some of predominantly black C to G mutations, some of predominantly gray T to A mutations, some predominantly of these green T to C mutations, and some predominantly of these pink T to G mutations. And then there are combinations. This signature has C to A and C to T mutations, and this one C to A, C to T, and T to C mutations. So in other words, there are a lot of mutational signatures and thus a lot of mutational processes contributing to the mutations that we find in cancer, and they are very diverse indeed in their nature. So we can then ask the question, how do these signatures operate? How are they found across the range of human cancers? And that's what's shown in this plot. Down the left-hand side, you've got the 30 mutational signatures, and across the top, the 40 different cancer types from which we have extracted the data. And where you see a green blob, that indicates that that signature is present in that particular cancer type. So there are some mutational signatures, and therefore mutational processes, that are essentially ubiquitous, operating across all cancer types. That's these two here, signatures 1 and 5. Indeed, actually, these two, which I'm not going to talk about anymore in the course of this talk, actually are present in almost every cancer sample, not only every cancer type. Then there are signatures, such as this one, which is present in about half of the cancer types, this one, which is present in 3 out of the 40 cancer types, and these 3, each of which is present in only 1 cancer type. So some of these mutational signatures are very common, almost ubiquitous. Others are very rare, found only in a single cancer type, and some of them, thus far, only in a single cancer sample. We can then look at each individual cancer type and ask the question, which signatures and how many are contributing to them. So for acute myeloid leukemia, there are just 2 signatures found. For ovarian cancer, there are 3 mutational signatures and therefore 3 mutational processes operating. For esophageal cancer, there are 7 mutational signatures thus far, and for breast cancer, which currently is in the lead, there are 12 mutational signatures that we found. So in breast cancer, there appear to be 12 mutational processes that are contributing to the somatic mutations found. So everything I've told you is a mathematical construct. They're all patterns. What we, of course, want to know is what is the biology underlying those patterns? In other words, what are the mutational processes underlying those mutational signatures? And I'm just going to pick on 2 of those mutational signatures to illustrate the search for mutational processes. And the 2 signatures I'm going to talk about are signatures 2 and 13, which are related. And you can see their relationship immediately because they are found in exactly the same cancer types. And as I'll show you in a moment, they're found together in the same cancer samples. I mentioned to you before that what the algorithm will do is it will not only extract the mutational signatures, it'll also estimate the number of mutations attributable to each signature in each cancer sample. And that's illustrated here. Here there are about 30 to 40 breast cancers of about the 1,000 or so that we have analyzed. The height of the bar tells you the number of mutations per genome in each breast cancer. So this is a breast cancer which has a lot of mutations in its genome, almost 90,000 substitutions. And at the other end, we've got a breast cancer with very few mutations, a few hundred mutations. So this is a selection of the breast cancers. The colors indicate the contribution of each of the signatures. And you can see two things from this plot. First of all, the mutations in each individual breast cancer are usually contributed to by multiple mutational signatures, because each bar has got multiple colors in it, and that there are multiple different combinations of mutational signatures that you're seeing there, because you're seeing multiple different combinations of colors. So there is tremendous complexity that is emerging in the contributions of mutational processes to individual breast cancers. So that's all the signatures that we found in breast cancer thus far. Let's concentrate for a moment now on the breast cancers that have got signatures 2, which are represented in red, and signature 13, which are represented in purple. And you can see them here. These 4 breast cancers have got a lot of mutations, as you can see by the height of the bar compared to the ones down here. And they have a lot of red, signature 2, and purple, signature 13 mutations. And that is very characteristic of these 2 mutational signatures. In other words, in many cancers, you don't find them at all, so you see no red or purple here. So in a lot of cancers, you don't find them at all. But in the cancers that you do find those mutational signatures, there are huge numbers of mutations attributed to them. They are hypermutational mutational signatures. What do those signatures look like? Well, this is signature 2. You can see it's characterized mainly by red C to T mutations, but you can also see that those C to T mutations are in a particular subset of sequence contexts. And those sequence contexts are where the mutated C has a T immediately prior to it. So there are TCN trinucleotides. So that's signature 2. Signature 13 is characterized predominantly by black C to G mutations, but you can see that these also are in particular sequence contexts, and these also have a T immediately before the mutated C. So both of these, so signature 2 is C to T with a TCN trinucleotides, signature 13 is C to G at TCN trinucleotides. So whenever we find a mutational signature like this, our first recourse is to ask, is there anything similar in terms of pattern found in the literature? And for these 2 mutational signatures, indeed there was. And if there is something found similar, that suggests that at least presents the possibility that whatever is causing that mutational pattern that's been found in the literature previously might be causing the signature in cancer too. So these 2 mutational signatures resemble the signatures of mutations in experimental system produced by this family of enzymes, AID and the APOBEC family. And these are enzymes which deaminate cytosine. The family includes AID, APOBEC1, APOBEC2, a whole family of APOBEC3s, and APOBEC4. The way that these enzymes work is as follows. The enzyme comes in, deaminates cytosine to generate uracil. Now, if DNA replication takes place at this moment, then it'll treat the uracil as thymine, and so ultimately we will end up with a thymine there and get a C to T transition. There's the C and there's the T. But of course, uracil shouldn't be in DNA, and that will be sensed by the DNA repair machinery. and it will be removed, leaving an A basic site. And if DNA replication takes place now, the replication machinery is going to have to guess what to put in across that A basic site. And the set of enzymes that do that will often guess to put in a G. So you end up with C to T and C to G mutations, which is exactly what we found in those two signatures, signatures 2 and 13, and which is the reason why we hypothesize that the APOBEC enzyme system is behind those signatures. What do these enzymes do? Well, you're probably very familiar with them. AID plays a central role in somatic hypermutation and class switch recombination at the immunoglobulin loci. Essentially, it is responsible for generating the diversity in immunoglobulin genes. The APOBEC enzymes have a rather different function. They're part of innate immunity rather than adaptive immunity. And they are thought to be, at least in part, there to respond to invaders, to viruses that get into cells, HIV and hepatitis B, for example, and also to influence the lives of retrotransposons when they jump. So the idea is when the virus comes in or the retrotransposon jumps, these enzymes get switched on and they mutate the DNA of the virus or the retrotransposon, trying to disable it, trying to inhibit it from going about its business. So they're part of the innate immune system. Here are the members of the family, and we can ask the question, which of these members is likely to be actually operating in generating the mutations of those signatures 2 and 13 in cancer? Well, it's unlikely to be the best studied of these, AID, because we know that the signature of AID mutations does not resemble that signature of 2 and 13 that we find in cancers with the T immediately prior to the mutated C. But it could be either APOBEC 1, 3A, or 3B, and actually the answer is we don't really know. We don't really know, but although there have been suggestions that it's APOBEC 3B, but we really don't really know. A good question is, why is this happening? I've shown you that some breast cancers and other types of cancer have huge numbers of these mutations attributable to these two signatures, and so something must have switched on these APOBEC enzymes to cause all those mutations. Is there mutation or amplification of the APOBEC genes? If there is, we can't find it. Is it because there's overexpression of the enzymes? Well, there is a little bit of correlation between overexpression and presence of lots of mutations, but it's a pretty weak correlation. It can't really account for the fact that we're finding such huge numbers of mutations attributed to those two signatures in certain subsets of cancers. A more interesting hypothesis is that if we find these massive numbers of mutations attributable to these two signatures, could it be because sometime in the past, or even still, that a virus has entered a progenitor cell to the cancer, or a retrotransposon has jumped, or inflammation, which can also switch on the APOBEC system, that somewhere in that cellular lineage, one of those things has happened which has caused the APOBECs to be switched on, causing loads and loads of mutations, and we end up seeing those in the final cancer. Could that be what's happening? That would be quite interesting because it would be suggestive that all these events could be etiological factors in the genesis of cancer. What's the evidence for that? Well, initially, there looked like there might be a little bit of wind in the sails of that hypothesis. Here I'm showing you about 30 different cervical cancers. Again, the height of the bar tells you the number of mutations per genome, and you can see in red signatures 2 and 13, and there's a lot of signature 2 and 13 in cervical cancer, and of course almost all cervical cancers have human papillomavirus in them. So this would fit with the idea that there's a virus in these cancer cells, and that's switching on the APOBECs, and they're producing all these mutations. This would fit. This doesn't fit quite as well, because in liver cancers, most of which in this series have got hepatitis B virus in them, you can see there is no red at all so there's no signature 2, and there's none of this light purple either, so there's no signature 13. So almost all these cancers have got hepatitis B, but they've got no APOBEC activation as far as we can see. So it's an interesting hypothesis, but it is as yet unproven, and the reality is that we don't know at the moment what is switching on the APOBECs in cancers to generate all these mutations. So everything I've been telling you about until now is mutations which are found right across the genome. They're raining down everywhere in the genome. I'm going to briefly touch on a slightly different, but as you'll see, related process, which is that of localized hypermutation, which we call kataegis. Kataegis is the Greek for thunderstorm. And to demonstrate kataegis, I'll show you plots like this, which are called rainfall plots. Now, this is data from one single breast cancer. And each of these dots is a somatic substitution, somatic mutation in that breast cancer. And they're colored according to this color code here. And the dots are organized such that on the left-hand side, you're at the top end of the genome, at the top end of short arm of chromosome 1, and as you move across, you get to the end of the long arm of chromosome X. Now, in this particular breast cancer genome, there are about 3,000 mutations. The genome is about 3,000 million base pairs in length. So if the mutations were raining down randomly in the genome, you would expect to find them about once every million bases. And what I've forgotten to tell you is that on the y-axis, what the y-axis is the distance between each mutation and the previous one in that walk through the genome. So this particular mutation here, for example, is only one base pair away from the one previous to it in the walk through the genome. And all these mutations up here are a megabase away from the previous one in the walk through the genome. So what this represents is random mutagenesis, mutations raining down in a completely random manner through the genome, an uninteresting genome. This is a more interesting genome because what you can see here is there's a set of mutations that are very much closer to each other. All of these are less than 100 base pairs away from each other, clustered in one particular part of the genome. And this is kataegis. This is the thunderstorm of mutations in a particular part of the genome. So that's one breast cancer with kataegis. Here is another breast cancer with kataegis. In this particular case, you've got lots of trickles of kataegis throughout the genome. And when you look at many breast and other cancers for kataegis, what you find it's never in the same place twice. This is something which starts apparently nucleated randomly in different parts of the genome. There's no particular part of the genome which is afflicted by kataegis. So that's the overall pattern of kataegis. What you can see is that the colors are different for the kataegic mutations than the randomly occurring mutations. So the kataegic mutations are red, which is C to T, or black, which is C to G, compared to the color mix that is up here of the randomly raining mutations. What you can't see, because I can't depict it on this portrayal, is that the C to T and C to G mutations are all of them at TCN trinucleotides. In other words, there's a T before the mutated C. And this may ring a bell for you because this was exactly what we saw previously when we were looking at the whole genome for signatures 2 and 13. For signature 2, C to T mutations at TPC context, and for signature 13, C to G mutations, where there's a T before the mutated C. So, if we made the hypothesis previously that APOBECs were responsible for signatures 2 and 13, raining mutations down right across the genome, could APOBECs also be responsible for kataegis? And actually, this is relatively straightforward to address, because if you just express APOBEC enzymes in yeast, yes, you get the mutations across the whole genome, but you immediately get these clusters, these foci of kataegis, without doing anything else at all. You get them happening. So the APOBECs clearly can generate these kataegic foci. The next question then is, why? What makes a position in a genome where a kataegic foci occurs? So what directs an APOBEC to a particular part of the genome to generate kataegis? And one possibility, which we tested, was a double-strand break. Randomly occurring double-strand breaks, could they be generating kataegis? And indeed, if you introduce a double-strand break into yeast, you immediately nucleate kataegis around it with those mutations. And this also makes some sense, because when you go back to cancers, what you find is that the kataegis, these clusters of base substitution, are very closely associated with clusters of genomic rearrangements. And of course, genomic rearrangements must occur after double-strand breaks. So our hypothesis here is that double-strand breaks occur in cancer genomes. Around the double-strand breaks, you get the kataegic machinery brought in for reasons that we don't understand. And when the double-strand breaks are repaired to make rearrangements in the genome, you end up with genomic rearrangements associated with these clusters of kataegic substitutions. Actually, kataegis is quite common. A little bit of kataegis is common. In breast cancers, more than half have a little bit of kataegis, and that goes for a lot of adult tumors. For childhood cancers, there is less kataegis found. But it's actually a pretty common phenomenon. So I've shown you how we can investigate the biology behind two mutational signatures. I'm now going to briefly review what we know about the rest. So there are about 30 of them. This one here is C to T, is predominantly C to Ts at CpG dinucleotides, which is, we believe, due to deamination of 5-methylcytosine, a very commonly understood process. This signature is the signature we believe is left behind by tobacco mutagens. This one is the signature left behind by ultraviolet light exposure. This one is the signature left behind by aflatoxin exposure. This is the signature left behind by this stuff, aristolochic acid. This is a dietary supplement that is an extract of plant that is used in a number of different parts of the world. This produces absolutely wild mutagenesis in the cancers that it's found in. And if there's anybody taking that here, I would suggest they reconsider. This is the signature left by the anti-cancer drug temozolomide, which is used in gliomas and malignant melanomas. So we found this signature in cancers which had been treated and had recurred after drug treatment. These are the signatures. There are 4 signatures associated with defective DNA mismatch repair. We don't understand why there are 4, but there clearly are 4. This rather featureless signature is the signature left by defective BRCA1 and BRCA2, in other words, defective homologous recombination repair. This was a bit of a surprise. We knew that defective BRCA1 and 2 cause genomes to rearrange in large structural changes. We didn't know that there was going to be a base substitution signature associated with absence of BRCA1 and 2 function, but indeed there is, and this is it. This is a signature associated with mutations in polymerase epsilon. These are the signatures of the APOBECs that I was showing you earlier, and these are the signatures for which we do not understand the etiology. How are we going to find those? Well, we can associate a signature with other aspects of the cancer, with epidemiology, with the expression of genes, with mutated genes. That may tell us what these signatures are due to. Or we can generate a compendium of signatures due to known exposures in experimental systems. And both of these approaches to understanding the etiology of those signatures is underway. If you want to know more about those signatures, you can look on the COSMIC database. There are now some pages dedicated to them. And in summary, there are about 30 genome-wide mutational signatures across human cancer. 2 of them that I've focused on in this talk reflect cytosine deamination due to APOBEC activity, and they're common. Kataegis is a form of localized hypermutation. It's found in many cancers and it's also probably caused by APOBECs. Some signatures are known due to known mutagenic exposures or defects in DNA repair, but the cause of many is unknown. And obviously, understanding the mutational processes underlying these mutational signatures will inform on cancer causation, potentially on cancer prevention, and even actually on cancer treatment. So most of this work is due to these two protagonists, Irina Nixanel and Ludmil Alexandrov, both of whom were graduate students with me and both of whom have their own groups now. A number of collaborators. A lot of the data was from the International Cancer Genome Consortium and the Cancer Genome Atlas. This is Sanger at Dusk, and thank you for your attention.
Thank you, Mike. I'll just take two questions.
That was very exciting. I wonder if you could just comment on whether it's possible to distinguish the somatic mutations that might be causal for the cancer versus those that happen after the transformation.
So we can identify those mutations which we believe to be causal by sort of mathematical approaches that are different from the ones I've shown you here. Some of the mutations that are not causal will have occurred before those causal mutations, and some of the mutations that are not causal will have occurred after the causal mutations. And we can actually separate both of those out to some extent by looking at mutations that are present in the root clone of the cancer and those that are subclonal. So one can do all those sorts of analyses and one can apply them to the signatures as well. Yeah.
It's very, very exciting talk. So if these signatures are due to specific causes, in some cases you said, okay, the organ is specifically exposed, such as with tobacco, but in other cases the whole organism is exposed. Have you looked at the signatures in biopsies or blood or in other tissues, in not non-cancerous tissues, in the same patient?
Well, that's a good question, and we haven't. The barrier to doing that is that the only reason we can actually do any of these analyses is because the cancer is a clone. So we're looking at mutations that are present in basically one clone. When we go to normal tissues, we immediately get into the position where if we take even a very small piece of normal tissue, we're looking at many, many clones, and it's quite difficult to find the mutations in them when you've got so many different clones. However, we have recently, about 4 or 5 weeks ago, published a paper in Science where we looked at these little core biopsies of skin. And we were able, this was of not cancer, this was normal skin from the eyelid, made tiny little core biopsies through it and then sequenced them. The biopsies were small enough that there were a relatively small number of clones in them. And we could see very clearly in those clones, all those clones in the skin were heavily mutated with ultraviolet light type mutations. In fact, the number of mutations that you find in normal skin due to ultraviolet light is no different from the number of mutations you find in skin cancers. So it's not a question of a particular cell having more mutations, it's just which genes those mutations fall into that determine whether that cell becomes a cancer.